we also start neuroanatomy on Monday, and I posted 3 of the neuro lectures, and we'll be posting the 4th probably on Monday. So if you're not doing anything this weekend and you want to get a jump on what's to come, you start flipping through those slides. Congratulations, if you didn't hear me yesterday, on making it to the halfway point. It's a big feat, no doubt, so congratulations.
Thank you.
Sorry about Brock's sunburn. Anyway, so some of the things that I'm going to start to talk about, Professor Polk will probably cover when she does neurophysiology, but I just need to make sure that they're covered, and that'll get us into today's discussion. There are a lot of objectives, but remember this is a 2-part sermon. When you look at the neuron and consider how many there are, and the numbers are there for the brain, the spinal cord, and the enteric nervous system, which is the part in the gut, the enteric nervous system. I mean, 500 million neurons in the gut lining. That's, or between the gut muscle layers. That's truly impressive. Truly impressive. And they all talk to each other, supporting the functional unit here. Will be the glial cells, and there are several types of glial cells I'll cover here in just a minute. But again, 10 times more glial cells than nerve cells. Myelin is a substance that's ubiquitous in the nervous system, both the central nervous system and the peripheral nervous system and the autonomic nervous system. Everything is ensheathed in myelin. You describe those axons as myelinated and unmyelinated or non-myelinated. Unmyelinated is the word I always use. But I just said that everything is ensheathed in myelin, and I'll show you why that is here in just a second. The other point you need to know about the histology of the neuron are when you look at myelin-forming cells in the central nervous system, the cell that has that function is called an oligodendrocyte. You'll see it in a minute. The cell that has the same function in the peripheral nervous system is called a Schwann cell. An oligodendrocyte will provide myelin to several axons. A Schwann cell will provide a myelin sheath around only one, a part of only one axon. So there'll be dozens and dozens and dozens of Schwann cells lining the axons from over a length. Okay? The pieces and parts to a neuron, you remember from anatomy. You're going to have the dendrites, and most of the cells that we're going to talk about will be multipolar. So you'll have many dendrites, the cell body, and then at the apex of where, or at the junction of where the cell body meets the axon is a clear area called the axon hillock. And then you have the axon, and a cell will only have one axon, but that axon can branch, in some cases thousands of times, to cover a lot of distance or a lot of targets. Now when you stain neurons, what stains is a dense substance called Nissl, N-I-S-S-L, Nissl substance. The axon hillock is an area where there is no Nissl substance, and that's what gives it a pale look to it. Functionally, it's not really anything that you need to worry about. It's just a histological feature of neurons. I'm sure that Professor Polk will go over propagation of an axon. from its receipt in the somewhere up here, across the membranes and down the axon. I'm not going to spend any time really on dealing with that, other than focusing on the synapse here in just a minute. When you look at myelin, let's go back to myelin for a second. There are different fiber types, depending on the size of the fiber and the information it conducts. This would be considered a myelin fiber. A myelinated axon right here. You take a Schwann cell, and during development, that Schwann cell will wrap around the axon and then start folding on itself so that you have what appears to be layers and layers and layers of cell membrane and cytoplasm from the Schwann cell. As the axon or the Schwann cell matures, the cytoplasm is extruded so that what you have up here, if you I don't know if it shows up on your computer screen or not, but what you have here is the axon, and you'll see lamellae, concentric lamellae of the Schwann cell forming the myelin wrap. You can see a little bit of the cytoplasm of the Schwann cell right here, but there's no cytoplasm you can discern between the wraps of membrane. Here's the rest of the cell out here. This represents an unmyelinated fiber. And this gets back to my conflicting statements earlier. This is a oligodendrocyte or Schwann cell, it doesn't matter, depending on where it is. You'll notice that these are the axons right here. The axons are wrapped by one wrap of the oligodendrocyte or the Schwann cell. They're not wrapped in lamellae, it's just one wrap. So here you have in this one cell, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 axons within that one cell. Now, this is one thing I want you to pay attention to. If I go back, you need to appreciate the size here. You see that mitochondrion right there? The mitochondria. These little speckles in here, by the way, are called neurotubules. Neurotubules are proteinaceous structures where synaptic vesicles made in the cell body travel down these neurotubules to get to the axon terminal. Right. But that's a mitochondrion right there, because the axon in the synapse have to have energy. The size of that mitochondrion and these axons here is the same. So these axons are tiny in comparison to the large myelinated fibers. These would be, an example here would be the unmyelinated fibers of the autonomic nervous system, or fibers that transmit, they're called C fibers, that transmit pain and temperature. They're called unmyelinated. They still have a myelin wrap, but it's only one layer. And that's the answer to that conundrum. If you look at this in my microscopic slide here, the axon is that thin filament. It's a very thin filament right there. You can see another one right there. And these big blobs are the myelin wraps. Myelin, by the way, is essentially a fat. And it appears in gross sections to be white. So when you say white matter, you're thinking of a bunch of myelinated fibers. So you can see the separation of the Schwann cells here. Now, of course, you know about the nodes of Ranvier. Anybody on this planet not know about nodes of Ranvier? Right? Everybody knows about Does everybody on the planet know about saltatory conduction? If you don't know about saltatory conduction, this crap's gonna show up on a test. If you don't know what it is, you better raise your hand. Everybody knows what saltatory conduction is?
I don't know how that is.
Okay, that's what I want, that's what I want, some honesty. There you go. So,
Thank you.
there are two ways to get,
I understand.
there are two ways to get information down an axon. And maybe I better back up just a second.
Yeah.
If we take, this is a little physiology. If we take the axon and propagation of an action potential involves ordinarily you have differences between the electric potential outside the axon versus inside the axon. Inside the axon is actually, if you've measured it, it's actually at about, depending on the axon, about minus 60 millivolts. All right, just follow me here for a second. If I want to get an action potential moving down here, what happens is channels open up, sodium, which is a positively charged ion, pours in, and you change the membrane potential so that it's more positive inside the cell. And these channels, they keep opening up in a domino type effect. And this change in the membrane potential, it travels distance. So this charge, when you want to send an action potential, what you're doing is you're changing the membrane potential of the axon from minus 60 up to wherever it's going to set itself off to propagate that action potential. Okay? So, that's one way to get the information down the axon. Another way, somebody came up with this clever thing here, is what we're going to do is we're going to put these Schwann cells in the way, and saltatory conduction occurs not by movement like this, but by the change in membrane potential jumping from node to node. So instead of traveling down the membrane, it jumps over from node to node. And it speeds up the transmission of an action potential. And that's what, in a nutshell, saltatory conduction is. It speeds up an action potential. If you take a large myelinated fiber, an example of a large myelinated fiber would be what's called an A delta fiber. That's a large fiber. By contrast, here's the C fiber here. A large A delta fiber can propagate an action potential somewhere in the speed of about, if I remember right, now I'm having to reach back, way back here to come up with this, because I'm sort of making all this stuff up. About 30 meters a second. So it's very fast. You're only 2 meters tall, so an impulse can go from your big toe to the top, to your cortex here, in just milliseconds. C fibers, on the other hand, are slow by comparison. They conduct at about a couple of meters a second. So they're much slower. An example of this would be, you know, if I take Meredith's hand and I hit it with a hammer, you're looking at that thinking, oh no, here comes the pain, right? Here comes, and then ow! Okay, so here's the thing. you perceived what went on, right? It instantly went up to your central nervous system at 30 meters a second. You knew it was going to hurt before the C fiber information actually made it there. That's kind of an example of it. See what I mean? Okay. I'm going to be talking about a lot of stuff over the next week. If you don't understand something I say, stop me, okay? All right, let's look at synapses. Synapses, here's the thing. Nobody realized what a synapse was, or it was an actual gap between neurons until about the 1960s. Did you know that? That's amazing. It wasn't until the invention and use of the transmission electron microscope in the 50s and 60s that they actually realized that all these neurons were not connected, physically connected. There was a gap that separated them all. And that gap is a few nanometers wide here. A neuron does not touch another neuron. This is the synaptic cleft. And there are a bunch of synaptic clefts. 500 trillion. If you look at 1 cubic millimeter of your brain, 1 cubic millimeter, look how many synapses are in 1 cubic millimeter. Can you get your, I can't wrap my head around it. I actually did a study where we counted the number of synapses in spinal cord and came up with huge numbers like this. Try to wrap your head around the communication that has to occur when you're talking about hundreds of millions of synapses. You can't, it's impossible to even imagine that. The complexity of communication in your nervous system. Truly impressive. If you leave here today, there's one thing you need to understand. Truly impressive. All right. The elements of a synaptic junction. You've got a presynaptic membrane, so this is the axon terminal here. And then you've got the postsynaptic item. What that item is can be very variable. Synapses can occur directly on the body of the neuron, the subsequent neuron. They can occur on the dendrites. They have these little things. Dendrites sometimes have these little things called dendritic spines. They can occur there. They can occur actually on other axons. So you can have what's called an axo-axonic synapse. Now, why would you have an axo-axonic synapse? Well, here's the thing. Synapses, they release into the synaptic cleft the contents of the vesicles here, the presynaptic vesicles. The transmitter, or the substance in that vesicle, can be either excitatory to the next neuron in the chain, or it can be inhibitory. So something that's excitatory would cause this membrane potential to change towards 0, and you would propagate an action potential in the next neuron. Depolarization of the next neuron. However, if I took a substance like gamma-aminobutyric acid, GABA, what it does, it's an inhibitory neuron. How does an inhibitory neuron work? I mean, neurotransmitter work? How does an inhibitory neurotransmitter work? It takes the action potential and takes it lower. It makes it lower than -60. Therefore, in order for this neuron to fire, you're going to have something come along that raises it to a much greater height. That's simple. There are 45 billion excitatory neurotransmitters. There are 45 billion inhibitory neurotransmitters. They're not really, just a couple that are predominant ones. but you have, I mean, there are certain systems in your brain that we'll talk about. Well, I'll give you one, the cerebellum. The entire output of the cerebellum, and you know the cerebellum deals with movement, right? The entire output of the cerebellum is inhibitory. So there are certain systems in your brain that's their role. They inhibit, they shut down neural activity. And we'll talk about next week what happens when those systems don't work. All right. Any questions? Good. All right. Now, there's some issues that you need to know about synapses here. The nervous system is not static. It can't be static because a few years ago, I learned how to scuba dive. I didn't know how the year before that. I had to learn a whole bunch of stuff to pass the test and to do the movements that are associated with scuba diving. If my nervous system is hardwired, then how are you going to learn? Your nervous system has to be plastic. Plasticity is one function of synapses. You make synapses. Certain systems of your cortex can make new synapses as you learn new things or as you change. So there is synaptic plasticity in the nervous system. You're not growing new axons or new dendrites, you're just creating new synapses. What do those new synapses do? They strengthen the interaction between neuron A and neuron B. Synaptic strength. Synaptic strength is also a function of what's called LTP, long-term potentiation. If you have a synapse here onto a neuron here, the actual biochemistry of that synapse can change so that it takes less neurotransmitter to excite the postsynaptic neuron. Quantal release of neurotransmitter is what that's in reference to. Quantal release is like we're going to release so many picomoles of neurotransmitter. But in certain cases where you have LTP, the postsynaptic cell can change, the receptors can change to make them more sensitive to this neurotransmitter release. So it doesn't take as much to activate the cell downstream. And when the cell downstream activates, it stays activated longer. This is thought to be the mechanism for learning. Long-term potentiation is thought to be the mechanism involved in learning, synaptic plasticity. The more you use a memory, the easier it is to recall that memory. Let me give you an example. Raise your hand if you remember the name of your first grade teacher. You don't remember the name? You two? You are you two, the only people in class, just about. A couple of you. Okay? Almost all of you remember the name of your first grade teacher. Raise your hand if you remember the name of your fourth grade teacher. Still got quite a few of you. How about your seventh grade teacher? Homeroom. Homeroom. Not many of you. Not many of you. Why is it that almost everybody remembered the name of your first grade teacher, but not your seventh grade homeroom teacher? Who cares about your seventh grade homeroom teacher? Once you got out of the seventh grade, you never talked to her again or him again in your life. But you always thought about your first grade teacher. You kept going back to that memory. You've kept going back. I can tell you, my first grade teacher, that was 48 years ago. No, 50, a long time ago. It was over a half a century ago. I can tell you the name of my first grade teacher. That's
I can
it.
tell you I had a cool haircut. I had a mullet.
You still have a boy. Anyway. All right. Neuromodulators. Neuromodulators are in synaptic vesicles at the end of the terminal, but they don't actually cause an action potential of the downstream neuron or the muscles, the axon, or the environment, whatever the downstream thing. They don't cause it to fire. What they do is they modulate the membrane of the downstream target to either make it more susceptible or less susceptible to excitation. They modulate the downstream target. So when you look at certain systems, you might have one neuron that's going to release acetylcholine, but also it has synaptic vesicles in the terminal that contain peptides like substance P, bradykinin, cholecystokinin, somatostatin. You don't need to write these down. What I'm just saying is that these are all neuromodulators that they have an action in the small environment there. So they modulate the post or downstream excitation. Let me give you an example. SEAL training, military SEAL training, right? These people undergo repeated trauma during SEAL. Did I say that wrong?
No, I was thinking like seals.
Oh, not that I like SEAL, Navy SEALs. Good point, though. So they undergo intense training that's physically painful. And they do it so many times that over a period of time, they become immune or tolerant to the pain. They modulate their response to neurotransmission of pain. That's an example. Almost all systems in the nervous system have some type of neuromodulators. The thought that you've always grown up with is that one axon synapses on another axon, and you get an action potential. You forget that. That ain't how it works. It's much more complicated than that. Okay? Why is it that if I hit Meredith on the head with a hammer, she responds one way, and I come over here and hit Kayla on the head with a hammer, and she responds a completely different way? Their responses to pain are different. They modulate it differently. Okay? Synaptic density. Synaptic density, there's actually three things about synaptic density. One is how many synapses are associated with something. If you take the cerebellum, the primary cell of the cerebellum is called a Purkinje cell. P-U-R-K-I-N-J-E. Purkinje cell. These are enormous cells, very large, and they have a huge dendritic tree. So you have a Purkinje cell here, you've got one axon, right? That's gonna leave the cerebellum or get input out of the cerebellum. And a Purkinje cell has a huge dendritic tree. They're really pretty cells. Okay? There are two types of input to a Purkinje cell. One fiber type, so if we've got here Purkinje cells along the way here. One fiber type comes in and makes one synapse on one Purkinje cell at a time for a total of about 10 Purkinje cells. Another fiber type comes in and only goes to one Purkinje cell, but makes up to 1,000 synapses on that one cell. This is an example of spatial versus, well, let me put these. To excite a great big neuron, it takes a great big input. One synapse firing to this big neuron is not going to cause that, it's not going to change enough membrane the ion flow to cause that post cell to fire. One axon's not enough to make that thing fire. But, I mean, one synapse is not enough to make that big neuron fire. What you have to have is you have to have a whole bunch of synapses hit that neuron at the same time. That's called temporal summation. So if you take this system here, this one right here, you've got this, so we've got our voltage here, right, there's 0. We've got our voltage here, we're starting here, this one fires, bring it up a little bit. This axon, this synapse fires, bring it up a little bit. That one, down. So what you do is you get a stepwise approach until the whole thing fires. Bam! And you have an action potential. So, one fiber synapsing so that the temporal time is at the same. It fires. Now for the other type of input here, what you're going to have in order to get that neuron to fire, you're going to have a whole bunch of these, a whole bunch of stimulation at the same time. So the impulse to fire these Purkinje neurons originates here. Okay? Spatial versus temporal summation. I don't know if I said that right a few minutes ago because I'm getting kind of wound up. Spatial versus temporal summation. And that's how your nervous system functions. Every system, just about every system in your body, neural system functions that way. It's not one axon, synapse, the next axon in the chain, like I taught in neuroanatomy, right? Everything was connected, pre-ganglionic, post-ganglionic. No, it doesn't work that way. This is the way it all works. In order for something to happen, you've got to have enough of an input, either all at once or a big input, to make it happen.
So the big input is spatial.
Yes. Spatial, temporal. Temporal time. All the synapses happen at one time. Spatial is space. A whole bunch of neurons. So what does that mean? So here's the bottom line, what does that mean? If you're talking about this input here, the output is going to be very, very specific. If you're talking about this input, since you're talking about a whole bunch of Purkinje cells, the output is going to be more general. I'll give you an example. The eye movement muscles, extraocular eye movement muscles. There's one neuron that goes to one muscle fiber. When that one neuron fires, that one muscle fiber contracts. The nerve that goes to your latissimus dorsi, what nerve is that? What? Did you say it? Thoracodorsal.
Yeah.
Thoracodorsal. There you go. That a single axon to the latissimus will branch and innervate 1,000 muscle cells. That's why your extraocular eye movement muscles can move very precisely, whereas your back muscles don't. You know, this movement is a lot different than focusing from your nose to the nares, moving your eyeball just a fraction. All right. We have 74 slides to go. Okay. One more thing about synapses here. When you get when the action potential reaches the nerve terminal, it causes the synaptic vesicle to bind with the presynaptic membrane. There is a receptor situation here. It's a docking protein on each side that causes them to bind. Now, you need to know, and you will learn, that you have receptors. Once the neurotransmitter is released, you have receptors, obviously, on the postsynaptic membrane. You also have receptors on the presynaptic membrane. If you dump out norepinephrine into the synaptic cleft, it's going to bind to an alpha-1 or a beta-1 receptor, and you'll learn these receptors in physiology. But in the case of an alpha-2 adrenergic receptor, it's on the presynaptic membrane that actually inhibits the release of norepinephrine presynaptically. So the receptor would be up here. Once norepinephrine is dumped into the cleft, some of it's going to bind down here, but some of it's going to diffuse over here and bind. It's going to shut down the presynaptic excitation. It's a modulator. Okay? It inhibits. The other thing here is that once you get neurotransmitter bound, you got to get it off of the receptor. Otherwise, it's going to continue to stimulate the downstream neuron. You get it off the receptor, or you eliminate the excitation either by enzymes in the cleft that will degrade it, or you reuptake the neurotransmitter back into the presynaptic terminal. You know what an SSRI is? Everybody knows what an SSRI is. Everybody's on Prozac, Zoloft. I mean, pick one of them. If you're not on one, you better go get on one. What does SSRI mean? Select serotonin reuptake inhibitor. What is it doing? Is inhibiting the reuptake of serotonin, thereby increasing the amount of serotonin available in the synaptic cleft. That's what an SSRI does. All right. This is a sort of the, what actually happens here. Remember we were talking about before what a membrane looks like. Remember you've got the phospholipid bilayer here? You got the phospholipid bilayer of the presynaptic membrane containing the docking proteins. You've got the synaptic vesicle, which is a bilayered membrane. I said almost everything was bilayered. One or two things are not, but this is. This has to bind with this. So it fuses with it. Here's my vesicle right here, containing neurotransmitter. And the tabletop is the presynaptic membrane. It comes along here and binds at the docking proteins and then splits and opens up and dumps the content into the synaptic cleft. You can understand, hopefully, that it wouldn't take long, many synaptic vesicles, before, if I'm adding all of this membrane to the presynaptic vesicle, what are you going to do with that? All of a sudden the synapse grows from that big around to that big around because you got all of this membrane being added to it? You got to get membrane out of the presynaptic area. And the way you do that is by endocytosis of membrane up here. So whenever you get membrane added, whenever a vesicle adds membrane down here, you have to pull it in up here. So that's going out, that's going in. Otherwise, this is just going to keep getting bigger and bigger and bigger. You got to get rid of that extra membrane. Or not get rid of it, you've got to draw it back in. Okay? What time did we start preaching?
Like 11:40.
Okay, we got a couple more minutes, then we'll take a break. Glia. I said there were 10 times more glia than neurons. There are a whole bunch of different glial types. The predominant glial type is the astrocyte. And there are two types of astrocytes. There are protoplasmic astrocytes and there are fibrous astrocytes. Protoplasmic astrocytes are found in the gray matter. Fibrous astrocytes you'll find in white matter. Oligodendrocytes we talked about, the myelin-forming cells. Microglia are tiny little boogers that have phagocytic properties. They eat debris that's within the CNS. By the way, these things are not found in the peripheral nervous system. These glia are only found in the central nervous system. The brain, the brainstem, the cerebellum, and the spinal cord. That's the central nervous system. And then you have cells that are called ependyma. Ependymal cells line the ventricles of the brain and the central canal of the spinal cord. In the peripheral nervous system, in addition to Schwann cells, the cell bodies are encapsulated by a little glial cell called a satellite cell. If you look at this histological section, here's a neuron. There's a neuron, there's a neuron, there's a neuron. You can see the neurons. All these little blue cells are the glial cells. Glial cells divide. One of the functions of glial cells is, especially the astrocytes, is that they have a connection between the blood vessels and the neurons. as illustrated in this section up here. And one of their functions is to transfer nutrients from the blood vessel into the neuron. And transfer garbage from the neuron back to the blood vessel. So glial cells not only provide a nutrition support for the CNS, they form a scaffold. They also, through the microglia, are the garbage collectors. This is an example of the types of transfer of chemicals between the glial cell and the nerve cell right here. Lots of amino acids. You don't, I just thought this was a cool slide. Other than that, we'll move on. All right. So any questions about sort of the cell biology of neurons? All right, we'll take a few minutes break and then we'll come back, okay?